Hey guys, welcome to 13.3 and 13.4. We are talking about meiosis, the process, and how variation applies to it. So let's first talk about what variation is and where it's introduced. And so if we look at all of these kittens are siblings, but we can see that they all look different. So they came from the same parents, but they all have different genes which make them look different, and they are adorable. And so it is these three processes that we're going to talk about that play a role in helping these kittens all look really different. The first one is crossing over. Crossing over occurs after interphase or during prophase 1. This is called synapsis and crossing over. So remember that it happens during prophase 1 only and not prophase 2. And one thing we need to know is that during meiosis, our chromosomes are going to line up a little bit differently. If you can see here in mitosis, we have 46 chromosomes that line up, let's say 46 deep, along the metaphase plate. But during meiosis, our homologous pairs line up. So our first chromosome from your mom is going to line up next to the first chromosome from your dad. And the second chromosome from your mom is going to line up next to the second chromosome from your dad. And so we are 23 chromosomes deep because they are paired up. And what happens, there's 4 steps for this synapsis and crossing over. The first step is that the homologous pairs, or the homologs, are going to associate with their pair. And so then the chromatin begins to condense. And then a zipper-like protein called synaptonemal complex, you can see that in the green here, is going to sort of sew the homologs together. And so they're sort of stuck together. This attachment from the synaptonemal complex is called synapsis. So they are going to be next to one another. During synapsis, the DNA breaks, and there's an open end. And so it's going to break at the same spot on the paternal chromosome as it does on the maternal chromosome. And they're going to swap. So they're going to, the maternal chromosome is going to attach over to the paternal, and the paternal is going to attach over. So this is called cross. They're going to cross over, and they're going to then reattach to each other. You can see a picture of that right up here. And so this is the crossing over aspect of this. And where they cross over, you can see that they are, when the 2 chromosomes pull together, you can see at the chiasma, or multiple chiasmata, they're going to be stuck together. They're sort of sewn together. And here you can see that these synaptonemal complexes are not the same as cohesions. Remember that cohesions are what sew the sister chromatids together. These homologs are going to be sewn together with the synaptonemal complex, and then they are going to break apart. The synaptonemal complex is going to break apart and move away, and at that point, this DNA is now considered recombinant DNA because it has recombined. It is reorganized, and so we call it recombinant DNA. So that's the crossing over. The second process is called independent assortment. And so like we said before, when we looked at the way that DNA is organized, or the chromosomes are organized during meiosis, we remember that the chromosomes, the homologs, are lining up next to one another. But we see here that all of the blue, so let's just call those coming from the father, are lined up next to the ones coming from the mother, and the mother is all on one side and the father is on the other side. Well, independent assortment states that this is not going to happen. What's going to happen is that they're going to assort independently. Sometimes you're going to have one from dad on the left, and sometimes you'll have one from dad on the right, and sometimes you'll have 2 in a row from dad on the right, and sometimes you'll have 3 in a row from mom on the right, and they're going to line up independent of what the other chromosomes are doing. And that's why they call it independent assortment. And so in this picture, you can see that there are only 3 chromosomes, so n equals 3. And that gives us, if we put 2 to the 3rd power, that gives us 8 possible ways for our chromosomes to line up. But if we have 23 chromosomes, so 2 to the 23rd, well, that's going to give us 8.4 million combinations of chromosomes, meaning maybe the blue will be here this time, and maybe the blue will be somewhere else the next time. And it's going to be 8.4 million different possible combinations. And so that's going to allow for 8.4 million different outcomes of a cell at the end, or chromosomal combination at the end. The last one is random fertilization. And this looks at meiosis and the process of fertilization. And it looks at it in a sort of bigger picture, say, looking at the combinations, the possible combinations that can come from all of the randomness in the process. So if we have 23 chromosomes randomly assorting, that gives us 8.4 million possible combinations. You guys know that. But if we take 8.4 million combinations from the male, and we take 8.4 million combinations from the female, so 2 to the 23rd times 2 to the 23rd gives us 70 trillion different diploid combinations. This is astounding, the amount of potential variation. The variation also is introduced by crossing over, so that includes even more. And then finally, if you think about all of the variation that can occur if the fact that you've got about 4 billion people, if there's 8 billion people on this planet, that means about half of the people on the planet are about 4 billion. And so you have many, many, many different people to choose from, to procreate with. And so the amount of potential, the amount of variation that is introduced in sexual reproduction due to meiosis is amazing. So let's look at prophase. Let's look at meiosis, the steps of meiosis, and where those 3 different processes fit in. And as we talk about these steps in meiosis, we're going to assume that you already know all of the steps of mitosis. And so we're not going to talk about all of the things that are happening from mitosis because that's assumed. So here in meiosis, we need to have HALVE, the amount of chromosomes, the number of chromosomes in each cell. And so we're going to start with 1 diploid cell, and we're going to end up with 4 haploid cells. And so in prophase 1, so that means we're going to start with 1 cell that has 46 chromosomes, and they have a sister chromatid, or an exact copy. And we're going to end up with 4 cells that have 1 copy of 23 chromosomes. So during synapsis, or during prophase 1, yes, all of the things happen, that the centrosomes move to the poles and the nuclear envelope is breaking down. But what happens that's different in meiosis than in mitosis is that synapsis and crossing over is occurring, and the chiasma appear. Everything else is the same as mitosis, as if it was cell division. And so now during metaphase, we think metaphase middle, our homologous chromosomes are going to line up along the metaphase plate. So we can see here that our homologs, 1 from mom, 1 from dad, are lining up. Remember, there's going to be about 23 deep, and they are going to be on different sides. And so this is when the independent assortment happens. So one from dad is going to be on one side, and the other from mom is going to be on that same side, and it's going to be all different. It doesn't matter where which way one set of chromosomes line up, because they all assort independently. And then the microtubules from one side are going to attach to the chromosome that it's closest to, and the centrosomes on the other side is going to attach to the chromosomes that it's closest to at the kinetochores, and then we're going to get ready for anaphase. Anaphase away. The separase is going to cleave the cohesions that are keeping the homologs together due to crossing over. And then we're going to see the homologs are going to move to opposite sides. The sister chromatid cohesions remain intact at the centromere, though. So we have, so we're going to have sister chromatids that are not identical because of crossing over. So we can see that this set of sister chromatids has a little bit of blue on one side and a little bit of blue on and a little bit of red on the other side. So they are not identical, but they are still sister chromatids. Telophase 1 and cytokinesis. So when telophase 1 begins, each half of the cell has a set of 23 chromosomes with its duplicate. So just like we said, we have 23 chromosomes with a sister chromatid that is not identical. Cytokinesis is usually happening simultaneously, and as telophase 1. In some species, the DNA will decondense and the nuclear envelope will reform. But please remember that no interphase. We're going straight into meiosis 2. And this is important because we're not going to replicate the DNA here. There is no DNA replication. We're not going into G1 and replication. We're going straight into to meiosis 2. So here we are, prophase 2. And once again, the same things happening that happens in prophase. The nuclear envelope's breaking down, the centrosomes are moving around, the spindle apparatus forms. In late prophase 2, the microtubules begin to move the chromosomes to the metaphase plate. And now during metaphase 2, if we remember that we now have 23 chromosomes, and they're going to line up at the metaphase plate, and they are going to be 23 deep. So we have 23 non-identical sister chromatids lining up during metaphase 2. Spindle fiber is going to grow, it's going to attach, we know all of this. And here during anaphase 2, anaphase away, the separase is going to cleave the cohesions at the centromere, and the sister chromatids are going to split. And now they become chromosomes, independent chromosomes, as they motor towards the poles. And this is anaphase 2. We are almost done. So telophase 2 and cytokinesis. So nuclei form, the nucleoli form, chromosome condenses, and cytokinesis occurs just like it normally would. And so what we have here is each cell has a haploid set of chromosomes. It has one copy of 23 chromosomes, and each cell is genetically distinct from one another and its parent. So each of these 4 cells is considered to be a sex cell, and if it's a female, it's going to go into a process called oogenesis, where 3 of these cells are actually smaller than one cell. One cell is going to have gotten a majority of the cytoplasm, and that will become the egg. And these 3 cells are sacrificed. If this is a male, then these become 4 sperm. And so each of those sperm are genetically different. So sexual reproduction provides a population with variation. Variation is really important at every level. Genetic variation in a human being makes that person a more healthy human being. Variation in a population is going to give that population a selective advantage to overcome obstacles when the environment changes. And the same is true with a community. If you want more different types of species within a community. So variation, diversity, is extremely important in life. And so within a population, if you have more organisms or more organisms within the population that are different, you're going to be given more tools to overcome obstacles created by a changing environment. So if we look at this picture, we have 3 different sizes, 3 different types of giraffes, one with a very short neck, one with a medium-sized neck, and one with a very long neck. And so this is variation within the species. And if trees grow to be taller and the short giraffes can't reach the leaves, well, then the tall giraffes can still reach the leaves. So variation in a population isn't going to save an individual, but it will save a population. It'll allow a population to adapt and not an individual. It's an important concept to understand because this short individual still isn't going to survive because it can't reach the leaves. However, the tall giraffes that can reach the leaves will survive, and so the species survives, but not all organisms survive. And if we look at some organisms that are that remain in stable environments, this, like the alligator or the crocodile, they haven't changed in many generations because they're very well adapted to their environment, and their environment is stable. And so there's no need for them to overcome changes in their environment. And so in these scenarios, it almost seems as though asexual reproduction would be a more advantageous solution because the alleles present in the population are well adapted for the environment, and it would make sense for those alleles to persist. However, if a change in the environment did occur, well, they wouldn't have the tools. They may not have the tools to overcome. They'd be less likely to have those tools to overcome a change in the environment. I hope this helps, and I will see you guys later.